Hello, everyone. Welcome to today's live broadcast, Translating Airway Gene Expression into a Biomarker for Lung Cancer and COPD. I'm Bob Woodard of LabRoots. I'll be your moderator for today's event. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem to the green Q&A button on the lower left. I would now like to introduce today's speaker, Avrum Spira. Dr. Spira is a professor of medicine, pathology, and bioinformatics and the Alexander Graham Bell Professor in Healthcare Entrepreneurship at Boston University. He is founding chief of the Division of Computational Biomedicine in the Department of Medicine at Boston University and director of the Translational Bioinformatics Program at Boston University's Clinical and Translational Science Institute. As an active clinician scientist, he is also an attending physician in the Medical Intensive Care Unit at Boston Medical Center. Please join me in welcoming Dr. Spira. I will now turn the presentation over to him.
Okay, thank you, Bob. Appreciate that. Again, I appreciate the opportunity to present. This very unique setting. I'll be spending the next 40 minutes or so telling you about some of our work on developing gene expression-based biomarkers to impact care of patients with lung cancer and chronic obstructive pulmonary disease, or COPD. Before I do that, though, let me get my disclosures out of the way. I'm a founder and consultant to a molecular diagnostic company called Allegro Diagnostics. That company was acquired about 6 months ago now by a publicly traded company in California called Veracyte. Veracyte has now launched a commercial version of one of our gene expression biomarkers for lung cancer detection that I'll be telling you about today. On the next slide is an outline of the three areas that I hope to cover over the next 40 minutes. I'm going to tell you how gene expression signatures in airway epithelial cells can serve as clinically relevant biomarkers to impact two smoking-related lung diseases. The first part of the talk is going to focus on early detection of lung cancer and how bronchial airway gene expression can impact that aspect of disease. I will say that's a very mature project. That molecular biomarker, as I mentioned, is already moved into the clinic, so it's a bench-to-bedside success story. The second bullet point is moving from lung cancer to another smoking-related lung disease called COPD. And the idea here is using gene expression in these same airway epithelial cells to help guide therapeutic strategies in that disease. And I'll tell you a little bit more about that in a moment. Last but not least, the third area that I wanted to cover is an emerging area within our group, which is to use airway gene expression as a measure of the physiological response to a variety of inhaled toxic exposures in our environment. And in particular, I'll show you one example with electronic cigarette smoke. But let me begin with the basic hypothesis that underlies all the work I'll be showing you today, the so-called field of injury. And this really is the basis for all of the biomarkers that will be presented. The concept is as follows. In individuals who smoke, even though diseases of the lung that develop, like lung cancer and emphysema or COPD, tend to develop deep within the lung parenchyma, all of the epithelial cells that line that respiratory tract, from your mouth and nose all the way down to your large airway epithelium, all of those cells are exposed to all the toxins in cigarette smoke, and they have genomic alterations as a result. So I'll put a little pointer here for this to work. So the idea here is that these cells have a certain genomic response to the exposure, and that how one smoker responds to the exposure and how it varies between smokers associates with risk for developing tobacco-related lung disease, most specifically lung cancer, but as I hope to show you today, also COPD. So if that hypothesis were true, then one could imagine developing a relatively simple tool to identify smokers at risk for having or developing lung cancer simply by collecting cells that line the upper airway, and again, we'll show you in a moment how we do that, and measuring the complete set of gene activity in these cells, and deciding is that a smoker on their way to cancer or not. So based on that relatively simple hypothesis, our group now has spent the last, I'd say greater than the last 10 years, working on developing an early detection strategy for lung cancer. And the reason that's important is shown on this slide. So many of you may know this already, but lung cancer is the #1 cause of cancer death in the world by far. And the major reason for that is we almost always detect it too late. By the time we diagnose it, it's often spread. And this is data from a Nature Cancer Reviews paper a few years ago. Shown in blue here are the percentages of these 4 common cancers in the United States that are diagnosed at stage 1. And what you can clearly see is lung lags well behind colorectal, breast, and prostate, with less than 20% of patients being diagnosed at the earliest stage. As a result, the 5-year survival for this disease, which is shown in purple, is dismal. Less than 1 in 5 patients will be alive 5 years after I give them a lung cancer diagnosis, and that number hasn't really improved in the last 3 decades. So what the data is telling us is that we in the clinical community are not doing a very good job of detecting this disease at its earliest and potentially curable stage. Now, when we talk about early detection of lung cancer, we're actually talking about identifying smokers who are at highest risk for disease, since about 90% of lung cancer in this country occurs in individuals with a history of tobacco smoke exposure. This is a very interesting graph. This was taken from a JAMA paper published last year, a study looking at the prevalence rates of smoking among adults across more than 100 countries in the world. And the color coding bar shows the darker red or darker the color, the higher the prevalence rate of active smokers among adults in that country. So in the United States, we're doing relatively well. Smoking rates have come down significantly as compared to a couple of decades ago. Roughly 19% of the adult population smokes every day, significantly lower than was the case 2-3 decades ago. But if you go to other parts of the world, specifically out in Asia and India, you see much higher rates. In fact, 65% of males in China actively smoke every day. And so there's a huge global burden on smoking. But the reality is not everybody who smokes develops lung cancer. So while 90% of lung cancer occurs in smokers, only about 10 to 15% of smokers, maybe as high as 20, will develop lung cancer in their lifetime. So the real question for us is how do we identify those smokers at highest risk for disease? And there was a huge development 3 years ago published in the New England Journal of Medicine that has changed the landscape for early lung cancer detection, and that was the National Lung Screening Trial, or the so-called NLST. And that was a study where they took 50,000 high-risk smokers and randomized them to either getting annual CAT scans of the chest as a screening tool for lung cancer versus a control group that got plain x-rays of the chest. And what they found, and this is one snippet of the data from that paper, they found that the group that got randomized to CT scans of the chest had a 20% reduction in their lung cancer mortality on follow-up, and arguing that CT screening can be a very effective way to identify those individuals at highest risk, identify the disease earlier in those people, and potentially reduce mortality from the disease. This is now effectively the standard of care. So based on that National Lung Screening Trial, the U.S. Preventive Service Task Force came out with a grade B recommendation in support of CT screening of high-risk smokers above a certain age with a certain smoking history, and now Medicare has agreed to reimburse for that. So we're now in the era of screening for lung cancer with CAT scans of the chest. The problem is we have 2 significant questions that we need to address in this setting. The first is that CAT scans tend to pick up a lot of abnormalities in the chest that are not cancer. And in fact, if you look at the National Lung Screening Trial data from the New England Journal paper, 1 in 4 patients who got screened with a chest CT had something abnormal concerning for lung cancer. Only 4% of that 25% ended up having lung cancer. So we need molecular biomarkers that can help us distinguish a benign from a malignant lesion found on chest CT. So diagnostic biomarkers are desperately needed, and I'll show you our attempt to do that today. The second need, which I won't have time to go into today, but I'll talk about at the very end briefly, is a need to identify who among the population of smokers, and there are about 45 million current and close to 50 million former smokers in this country, who among them should get screened annually with chest CTs. We don't know what the most effective way to use CT screening is, and if we had a molecular marker that could identify folks at risk for developing future disease, we could then stratify them into the CT screening surveillance. So this is a quick summary of 10 years of work in our group trying to address the two clinical needs on the previous slide, and we're doing that using gene expression profiles measured in bronchial airway epithelial cells. The way we get those cells is through a procedure called bronchoscopy, shown on this slide, where a fiber optic scope is inserted via the nose or mouth down through the trachea or down through the vocal cords and into the so-called trachea or windpipe. And we don't go very far in. We go in just after the trachea divides into a right and left main stem bronchus, shown over here, and then using a cytobrush through the working channel of that scope, we're able to rub up against the inside of that airway wall at that site and collect a relatively pure population of epithelial cells. And this is a key point. We're not biopsying the airway, but brushing it, and that allows us to get a relatively pure population of epithelium. We can then take those cells, extract the RNA, and run gene expression profiling using a microarray platform. And so back in 2004 and 2005, we started our studies on healthy volunteers, a group of active smokers and a group of never smokers who agreed to undergo bronchoscopy as volunteers. We got their airway epithelial cells and ran them on a microarray, and we found that active exposure to tobacco smoke changes the expression of hundreds and hundreds of genes and many microRNA in these cells. No major surprises there. What was a bit surprising was in 2007 when we went to former smokers, or people who had quit smoking. We found that most of the changes that occur when you smoke revert back to baseline within a few months of quitting, but a subset, about 20% of the genomic alterations persist years, in fact decades after you quit. So essentially there's an irreversible set of genomic damage in these cells. And that really set the stage for the third bullet point on the slide, and the clinically relevant one, which is identifying a gene expression signature in these cells that could distinguish smokers who get lung cancer from those who don't. And I'll be showing that's the biomarker moving into the clinic now, and I'll show you more about that in the next couple of slides. Just want to end the slide by the fourth point at the bottom left, which is that we recently published a paper together with a group at MD Anderson showing that these changes in the proximal airway, in the bronchial airway, which is not the site where the tumor arises, those genomic changes actually reflect genomic changes that are occurring in the tumor itself and the adjacent small airways. And I'll be showing you a little more of that data in a moment. But important data that connect the field of injury to the actual site of disease. But let me go back to the biomarker. This is a very busy slide, but it's meant to summarize what our biomarker is doing clinically. And so the question we're trying to address is one of the two that I alluded to earlier. Smokers, up in box one, smokers undergoing bronchoscopy for suspicion of cancer because of something abnormal seen on a CT scan or X-ray of their chest. The reason we do bronchoscopy as an initial test is it's a non-invasive, relatively non-invasive outpatient procedure. It's safe and has a very low complication rate, but the problem is the bronchoscope itself can't go all the way out to the periphery of the lung where many of these abnormalities arise and are seen on the CAT scan. So we go out as far as we can, and then we try and brush and biopsy and get cellular material back that's diagnostic. But we often fail to make a diagnosis at the time of bronchoscopy. And the sensitivity of bronchoscopy is anywhere from 30 to as high as 60 or 70% for diagnosing lung cancer. So very often we'll have a non-diagnostic bronchoscopy, and then we have a clinical dilemma, what to do next. Which of these patients should go for an invasive biopsy to be certain that this is not a cancer, and that's a biopsy that can be done by a needle through the chest wall, or an open surgical lung biopsy, versus who can we afford to watch non-invasively with repeat imaging of the chest every 3 to 6 months. That's really the clinical dilemma that we face week in and week out in our pulmonary practices. So what we did in the study was very simple. We told the pulmonologist, do all the routine studies at the time of your bronchoscopy, but before you pull the scope out of that person's nose or mouth, stop in the right main stem bronchus and just spend an extra minute or two and brush the normal appearing mucosa at that site. We'll take those cells, run them on a microarray, and the pattern of gene expression will tell us whether that abnormality on the CAT scan is a benign lesion or a cancer. So in the event that the routine studies the bronchoscopist does and collects are non-diagnostic, you would use the gene expression biomarker to decide this is a patient who should go to surgery right away, or this is someone who doesn't likely have cancer, and therefore we can avoid taking an unnecessary biopsy of that lesion. So if you go to box 5 on this slide, it's a quick summary of the work we published in 2007, where we identified an 80-gene expression signature in these cells that could distinguish a benign from malignant lesion on CAT scan. It had relatively good sensitivity. I just want to highlight that in 2 relatively small independent test sets, it had a very high sensitivity and high negative predictive value when you combine the gene expression biomarker with the routine results collected at bronchoscopy. So it had 95% sensitivity and 95% negative predictive value, which means that you can almost, not quite, but almost rule out the presence of lung cancer, which is what a clinician needs in this setting. They need to know who can they avoid sending for an unnecessary invasive procedure. So this is a biomarker that's moving into the clinic, and that's thanks to 2 very large clinical trials that have validated the biomarker called AEGIS-1 and AEGIS-2 on this slide. I involved 28 medical centers in Canada, the United States, and Europe, mostly in the U.S., as you can see from this map. More than 1,000 subjects were enrolled at 25 centers. All of these subjects are current or former smokers who are undergoing bronchoscopy for suspicion of having lung cancer. They had all the usual studies as per their standard of care, and then prior to the physician ending the procedure, they took an additional brushing from the main stem bronchus, the normal-looking main stem bronchus, and those cells were sent to a lab in North Carolina that ran it on a microarray and measured the expression of the biomarker genes. We then had to follow these people up after their bronchoscopy for up to a year until a final diagnosis was made for that abnormality on their CAT scan. And then we got to see, is our biomarker correctly predicting cancer status or not? So of the 1,000 patients, the first 600 were in so-called AEGIS-1 or a CLIA trial. Those results have been presented publicly in the last 12 months, and I'll share them with you today on the next slide. The AEGIS-2 study, those data will be presented next week at the American Thoracic Society meeting for the first time. But based on the successful validation in both trials, Veracyte has launched their test. The test that's not on this slide, but it's called Percepta, P-E-R-C-E-P-T-A. This is a gene expression-based assay for improving the preoperative diagnosis of lung cancer and reducing unnecessary invasive procedures. So here's the data on the next slide that's been presented at the American Thoracic Society meeting last year. This is the first trial, the AEGIS-1 study. What I'm showing you here is an ROC curve where we're comparing sensitivity versus specificity for our gene expression biomarker across the subjects in that first validation study. In dashed lines over here are all subjects in that trial, and the solid line next to it is that subset of individuals whose bronchoscopy was non-diagnostic. And that's important because that's the intended use population for the test. We want to, obviously if the bronchoscopy makes the diagnosis, you don't need the genomic biomarker, so we're really focusing our test on that subset of patients whose bronchoscopy is non-diagnostic. And the point here is they have roughly the same area under the curve of 0.78. The operating point on the curve, which doesn't project particularly well, so let me try and use the whiteboard here, is somewhere over here. It corresponds to a sensitivity that's close to 90% and a specificity of about 50%. But the most important parameter is the negative predictive value, or NPV. that is greater than 90%. So if you have a non-diagnostic bronchoscopy and a negative gene expression test, there's more than a 90% chance you don't have lung cancer. And that's what we believe will drive clinical utility of the test. It allows physicians to avoid unnecessary invasive procedures in those patients who don't have lung cancer. And so again, this was an important validation. Now, the test is moving into the clinic, as I mentioned, but one of the things that we absolutely wanted to evaluate was whether those changes we're seeing in the main airway next to the windpipe, the genomic changes, reflect those that are found deep within the tumor itself, deep in the lung. And so this is a slide summarizing a manuscript that was recently published in JNCI by Humam Kadara and Ignacio Estuba at MD Anderson that really illustrates that point. And what they did at MD Anderson is they took 20 individuals who had non-small cell lung cancer, early stage disease, who were having their tumor taken out or surgically resected. They took out the tumor and profiled gene expression in the tumor itself, but they also found small airways adjacent to the tumor, leading into the tumor, that they were able to brush and collect the epithelial cells from. And they ran those two on a microarray. And then they compared their data to our previous data set from 2007 that we had published looking at the main airway, not the small airways, but the large airway near the windpipe. And what they found is shown on the next slide, which is, I think, very important. They found a group of genes that were changing both in their tumor and adjacent small airways. So again, if I use my fluid over here, in dark green and light green are the two groups of samples that are behaving similarly for gene expression as compared to normal lung, which is over here in gray. And there's about half of those genes are upregulated in the tumor and adjacent normal airway over here, and about half the genes are being downregulated, or a little more than half, sorry, more than 3/4, I would say, are being downregulated. And if you look at those same genes in our prior data set from the Nature Medicine publication, and that's shown over here, you can see that those genes do a pretty good job of distinguishing the airway of smokers who have lung cancer, which is in green, from those that don't have lung cancer, which are in white, suggesting that at least a component of the gene expression changes in the proximal large airway reflect what's going on more distally in the lung, either in the tumor or in the airways right next to the tumor. So really connecting the compartments across this field of injury. Let me now pivot from lung cancer to COPD. So as I said, lung cancer has been a bench-to-bedside story. COPD is a more recent extension of the field of injury concept. And those of you not familiar with COPD, this is another smoking-related lung disease. Again, about 10 to 20% of smokers develop it. Unlike in lung cancer, the issue is less about early detection. This is a chronic, debilitating disease for which we have no good therapies, nothing to reverse the disease course. And so we were looking at airway gene expression as a way of measuring disease activity and response to drug therapy. And so we initially started our work back in 2013 here, where we extended that same field of injury concept to COPD. And we did that by collaborating with Stephen Lamb at the University of British Columbia. Stephen had a group of smokers who were being screened for lung cancer through bronchoscopy. And he has about 900 current or former smokers with and without COPD who've undergone that procedure, where we've been able to get brushings from their bronchial airway run on a microarray, and then look to see whether we can find a gene expression signature that distinguishes smokers that have COPD from those that don't. In our initial analysis that we published back in 2013, we took 87 subjects who had COPD and matched them 1 to 2 to subjects that did not have COPD, but had similar smoking history and similar demographic features. So the next slide is the bottom line from that study that was published in 2013. We identified a 98 gene signature that could distinguish airway samples from smokers who have COPD, who are in orange here, as compared to smokers without COPD, who are in blue. And I maybe should have mentioned earlier, but for those of you not familiar with these heat maps, each of the columns is a patient. Again, orange is a COPD patient, blue is a patient without COPD, and each of the rows is a gene. And blue refers to downregulation of a gene expression in a sample. and red is a higher level of activity of that gene in the sample. And you clearly see about half the genes are down in patients with COPD, and a little more than half are up. One of the interesting things about the genes that are up is we're able to find a transcription factor whose binding site is enriched in the promoter region of those genes, and that transcription factor is called ATF4. And why that's important is ATF4 is a very key mediator of the unfolded protein response. So when cells are undergoing ER stress, they elicit an unfolded protein response. And other groups have previously shown that that plays an important role in the development of emphysema, at least in animal models. And what our data is suggesting is that ATF4 may be driving the gene expression changes we see in the large airway of COPD patients. So to test that hypothesis, what we did is we took a bronchial epithelial cell lines in culture, and the cell lines we took are called down here are called Bs2B cells, and we overexpressed ATF4 and then did gene expression profiling 24 hours after the overexpression. And what we found is when you look up here at the genes that are changing in patients who are increased in expression in their airway when they have COPD, and that's shown in B. Blue are patients without COPD, orange are patients with COPD. This is a group of genes that are upregulated with disease. Those same genes are upregulated in cell lines where you overexpress this transcription factor, arguing that this transcription factor is in fact driving some of the gene expression changes that we're seeing in the airway with disease. The other really important point, I'll skip over this, the other really important point that we want to make is if in fact these changes in the airway can tell us something about disease activity and disease pathogenesis, perhaps we can use them then to understand better who responds to what drug in this disease, and maybe an intermediate marker of therapeutic efficacy. So our first stab at that was connecting with a study called GLUCOLD, shown on this slide. This is a large randomized, or relatively large randomized clinical trial in COPD with 110 smokers with disease who either received an inhaled steroid called fluticasone, that steroid plus a long-acting bronchodilator called salmeterol, or placebo, and then were followed for 30 months. And one of the interesting parts of that study is that the group in the Netherlands who ran the study actually collected bronchial samples from their airway for gene expression studies pre-treatment and then again at 6 and 30 months after treatment. And what we found is on the next slide, when we ran those samples on a microarray, we found that our 98 gene signature of COPD that I showed you earlier tends to revert back towards baseline in the group of folks who are in the treatment arms, either the steroid arm alone or the steroid plus bronchodilator, arguing that what we have here is the dynamic field that may in fact reflect response to therapy, and that's an exciting observation. These are 3 box plots showing you 3 of the 98 genes. The expression of those genes is on the y-axis. On the x-axis are the groups. So to the far left for each box plot, and I'll put a little arrow there, are the brushings collected from the smokers in Vancouver with and without COPD. And so this first gene, dual-specific phosphatase 5, is increasing expression at baseline in the airway of COPD patients. If you then go to the GLUCOLD study, which is to the right, you can see in the 2 treatment arms, the fluticasone arm or the fluticasone plus salmeterol arm, that gene goes back to baseline in terms of its expression by 30 months, whereas it doesn't change in the placebo arm. And the same is true of the next 2 genes on the slide. So arguing that these genes, in fact, are reversible, these alterations are reversible in the field with treatment. And that led then to our next paper in 2014, where we showed that the degree to which these genes change in response to inhaled corticosteroids in the airway represents or reflects how well the patient does in terms of improvement in lung function with treatment. So what we found in that study, again, this is the GLUCOLD study, we found that those individuals that have a stronger change in gene expression in response to the drug, they tend to have better improvements in their lung function, a measure called FEV1, and that's over here, as compared to groups who do not change their gene expression in response to steroids as much. And the other really important point, it wasn't just lung function, but in panel C, over here, it's quality of life. These are questionnaires that we administer about breathlessness, and the group that had a more robust response in terms of gene expression in red had a dramatic improvement in their quality of life in terms of their breathing. And so again, arguing that gene expression in the airway may be able to tell us in COPD who's going to be more likely to respond to inhaled corticosteroids, and that's an area that we definitely have an unmet clinical need. The last thing I'll talk about in COPD is a recent paper published last month in the American Journal of Respiratory and Critical Care Medicine, where we went one step further with this paradigm. We believe that COPD is not a single disease. We put a lot of patients under that umbrella, but the reality is we believe there's probably many different molecular subclasses of that disease, and essentially certain subgroups are going to respond better to certain drugs. And together with Stephanie Christensen and Prescott Woodruff at UCSF, we did a study where we looked at our gene expression changes in the airway of COPD patients and found similarities to a study they had done in asthmatics. And why that's interesting is on the next slide, because we know, and they have done a fantastic job in the past, the group at UCSF, of showing that a subset of asthmatics have a Th2 immune response active in their airway, a particular type of immune response that can be treated with drugs that target that pathway. And what we're doing here is looking at the 3 genes in that pathway, SERPIN-B2, CLCA1, and periostin, and looking at their expression across the airway of smokers with COPD, and we find a small subset, probably in the range of 10%, although it's not entirely clear, a small subset of those COPD patients have that pathway activated, and suggesting that they also have a Th2 immune response and that may be responsive to drugs that target that pathway. If we then go into the GLUCOLD study, we similarly find a subset that have that pathway active in their airway epithelium, and that subset tend to have a stronger response clinically to inhaled corticosteroids. The idea here is we may have uncovered a molecular subclass of COPD that might do better with certain treatments, and I really believe that is the future of personalizing treatment for that disease. I'm going to spend the last 10 minutes here on moving away from disease and moving the airway field of injury paradigm to understand response to exposure in the environment. And one of the things that's driving this is that we recognize that while our bronchial gene expression signatures, both for lung cancer and COPD, appear effective, they require a procedure to get the cells. And that's okay in certain clinical settings, such as the lung cancer diagnostic one, where you're doing the procedure anyway. But if we really want to move this to larger population-scale studies, we need to move out of the bronchial airway to a less invasive site. And we've been working with cells we can get from the buccal mucosa, shown here, as well as the nasal epithelium. It turns out the nasal epithelium is the more promising site anyway right now. We're still working in the mouth, but let me show you some of the data from the nose that is promising. Just to be clear, when we say we're brushing the nose, we're brushing the inferior turbinate of the nose. And that's shown over here. So if you press on your nostril and look up in the mirror, and I guess you can do that since no one's watching, you'll see a triangular protuberance on the lateral wall of the nose. Let me try to highlight it here a little better. That triangular protuberance is the inferior turbinate. It's about maybe a half a centimeter in from your nostrils. So what we do is just put a little cider brush shown at the bottom of the slide and rub up against the bottom of that turbinate. Spend 2 or 3 seconds and we get an incredible amount of high-quality RNA. We don't actually have to use any type of local anesthesia for doing this. It is non-invasive, and we've done this now on hundreds of adults and hundreds of kids. So with that approach, we then get a very good amount of RNA from the epithelial cells of the nose, and we can run it on a microarray. In our first studies back in 2010 with this approach, we showed that if you take healthy smokers and we bronchoscopy them and collect their nose and their bronchial airway on the same visit and run them in parallel on a microarray, that the gene expression changes in those 2 compartments mirror each other. In other words, genes that change in your bronchial airway when you smoke change it almost the identical way in your nose. And so it's a very effective surrogate. So we're now, that's of course healthy smokers. We're now moving into smokers with and without lung cancer and seeing whether the nose can be a surrogate for the bronchial signature that I showed you earlier. That would be exciting because then there would be 2 potential clinical applications, the 2 clinical needs I outlined at the beginning. One would be to, after someone has a chest CT shown over here and has something abnormal found, if you're not going to get a bronchoscopy, if the lesion is too small, too peripheral, or the patient is too thick, you potentially could basically brush the nasal epithelium and look at gene expression as a diagnostic marker. But I actually think the more exciting opportunity resides upstream of chest CT over here as a screening tool, where we can use this potentially on a population scale to identify which smokers are highest risk and therefore more likely to benefit from undergoing CT screening surveillance. Again, these are early days, these are ideas, and we have no published data to support this, but are trying to develop nasal gene expression biomarkers for these two specific clinical applications. And one of the ways we're doing that is through an initiative called DECAMP. This is a very large Department of Defense grant that we got 4 years ago now, that's allowing us to collect airway epithelial cells years before folks develop lung cancer and see whether, in fact, we can use gene expression in those sites to predict future risk of disease, which is what we would need if we want to demonstrate that we have a screening tool. This is a partnership between our group at Boston University and a group at the American College of Radiology Imaging Network called Akron. Akron is the group that ran over here the National Lung Screening Trial. They're partnering with us and a number of critical molecular biomarker groups across the United States, including UCLA, Brown, Vanderbilt, MD Anderson, to develop molecular biomarkers to help identify lung cancer early in military personnel and veterans who have very high risks of developing disease. We have 12 clinical sites that are recruiting patients and collecting samples, 4 active military hospitals shown at the bottom, 7 VA hospitals literally coast to coast, and 1 academic medical center. And there are 2 clinical trials that are actively enrolling patients right now. The first at the top is an indeterminate nodule study where we're taking 500 patients above a certain age with a certain smoking history who have an indeterminate nodule, generally speaking, between 0.5 centimeter, actually between 6 millimeters to be more correct, and 30 millimeters in size, or 3 centimeters in size. So this slide is incorrect there. Those patients are followed for up to 2 years until a final diagnosis is made, and we collect a variety of biospecimens, including airway cells, that allow us to look whether our biomarker and other biomarkers can accurately diagnose cancer. The second project at the bottom, I think, is potentially the more ambitious one, which is looking at healthy, or folks that don't have a nodule, but are above a certain age, have a certain smoking history, and either have COPD or a first-degree relative of lung cancer, and they're being screened for lung cancer and followed for up to 4 years for development of disease. And we're collecting all types of biospecimens at baseline and then annually, along with them getting screening chest CTs every year. And so the idea here is to try and identify individuals who are at risk to develop future lung cancer using a molecular biomarker in a number of different biospecimens. So again, that is how we're trying to move the nose and other less invasive sites into the screening setting. Last thing I'll say in the last 5 minutes is beyond lung cancer, the ability of the upper airway and the nose in specific to measure response to exposure might allow us to develop other biomarkers that are important for public health. And while I don't have time to show you today, we recently developed a nasal gene expression biomarker that can measure secondhand smoke exposure in children, which is a significant public health problem, and we don't have very good biomarkers for measuring exposure in kids to secondhand smoke. And we find that gene expression in the nose is a very sensitive tool for identifying which kids are being exposed. What I do want to talk about is the second bullet point, which is using the nose to evaluate alternative tobacco products, and especially with electronic cigarettes, which is a growing issue in this country. Last bullet point, which I won't talk about today, is this is actually occurring in the mouth, looking at gene expression as a measure of indoor air pollution, specifically smoky coal, in one of the provinces in China that has among the highest rates of lung cancer in the world. It's mostly in women who don't smoke, but instead are exposed to indoor air pollution 24/7 due to burning coal and heating their house and cooking and being exposed to the fumes 24/7. And so we're excited to try and expand the field of injury concept to measure response to a variety of exposures. Let me just show you the second bullet point, which is looking at alternative tobacco products. So one of the advantages of being less invasive in our sampling is we can now follow someone over time. We can sample someone's nose or mouth and repeatedly sample them as they change behavior. So this is one study that is not yet published, but has been done together with Steve Hecht and Dorothy Hatsukami, where at the University of Minnesota, where we've taken a group of smokers who are in a smoking cessation program, and we collect their nasal epithelium at time point 0, which is pre-quitting up here, and then we collect them at 4, 8, 16, and 24 weeks after they quit. We're running on a microarray, and we find a set of genes, and there's 101 genes in this heat map that change relatively rapidly within the first 6 months of quitting. We're highlighting in the box over here a subgroup of these genes that change extremely rapidly, change between the 0 time point and 4 weeks, so change within the first month. Majority of these 100 genes are changing within 4 to 8 weeks of quitting. Now, why is that valuable? So it teaches us a little bit about the biology and the kinetics of gene expression in response to quitting, but I think the more exciting idea is it provides us with a baseline that would allow us to evaluate folks who don't quit, but rather switch to an alternative tobacco product, and try to decide whether that switch, whether that product is in fact safer and causing less alterations to the genome. And so this is the paradigm we're trying to now use for electronic cigarettes. And that data is not yet published, and I don't have time to show it to you today. But what we're doing there essentially is having individuals who smoke regular tobacco switch to electronic cigarettes, which may in fact be a safer alternative. But what we're doing is profiling their airway gene expression pre and at time points after switching to see whether it looks similar to individuals who have quit smoking or not. And hopefully we'll have that data to publish very soon. So let me end there and just summarize what hopefully I've shown you today. First and foremost, that airway gene expression, specifically bronchial airway gene expression, can serve as a measure of the physiological response to cigarette smoking and potentially risk for developing lung cancer. We've demonstrated that it can serve as a diagnostic biomarker for lung cancer detection, and that is now moving into the clinic as a commercial assay. But we also decided to move this same field of injury paradigm to another smoking-related lung disease, COPD, where there we think it can serve as a therapeutic biomarker. There are two examples I showed you that today. One is where gene expression in the airway cells can tell you whether someone's responding to inhaled corticosteroids properly or not. And the second idea is it may help you identify, or us identify, novel molecular subclasses of the disease, so-called endotypes, that would allow us to personalize treatment for the disease. And that was the TH2 example we recently published. Now, last but not least on the slide is the idea that this field of injury might extend to the nose, and that site could be used as a less invasive surrogate tissue. And that would allow us to do a variety of additional things, including evaluate folks who are switching to alternative tobacco products and other inhaled exposures in the environment. I'll end there and just acknowledge a large list of individuals in this final slide, both at BU and outside of BU, that have been responsible for the studies I showed today, as well as the funding agencies at the bottom of the slide. So I thank you for your attention. Happy to take questions, I guess, either, online as this is done. So thank you again. And Bob, if I maybe hand it over to you now. Bob, are you there?
Yeah, thank you, Dr. Spira, for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question is from Konstantin Kurchev. I hope I pronounced that correctly, at the Universidad Autónoma de Madrid. In Spain, there's a risk due to radon exposure by the stones. They are epidemiological data confirming this hypothesis. Is it possible to detect earlier lung cancer due to the radiation?
Yeah, and that's an interesting question, and I think it reflects whether or not this field of injury concept that I outlined today is limited to tobacco smoke, which is in general the cause of lung cancer, more than 90% of lung cancer in this country, or whether it could extend to other causes of lung cancer. And you're absolutely correct that radon exposure in various parts of the world has been associated with increased risk of lung cancer through epidemiological studies. What's not clear is whether there would be a field of molecular injury throughout the airway epithelium. So it's a question that we don't have an answer to because we haven't studied it specifically. What I can tell you is we're beginning to look at other exposures other than tobacco smoke that lead to lung cancer and seeing whether they have the same field of injury. So, for example, the study I mentioned in China of non-smoking women who get lung cancer who have indoor air pollution exposure, where we are beginning to look at whether there's a field of injury there and whether that can predict lung cancer development in those women. Again, that's a very defined inhaled environmental exposure of biomass combustion or indoor air pollution. We don't yet know for other exposures like asbestos, radon. There's a variety of other environmental exposures associated with lung cancer. And our challenge, quite frankly, is getting access to airway epithelial cells for patients who have those exposures. And so if there are those out there that can access patients with those exposures, that's a very ripe area for collaboration with our group, and we'd be absolutely interested in studying that.
Okay. Are there any further questions? If there are, please submit them now. Well, I'd like to take this time to thank you again, Dr. Spira, and thank you all in the audience for attending. As a reminder, I'd like to let you know that today's webcast will be available for on-demand viewing for 6 months from this live webcast. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thanks again. We'll see you next time. Goodbye.